Dr. Abid Bhat. Yeah. So, Dr. Abid Bhat is a PhD in neuropharmacology from JSS Academy of Higher Education and Research in India. And he has completed a visiting research fellowship at Macquarie University in Sydney, Australia. And then he was a postdoc fellow here in University of Maryland, Baltimore. He published more than 20 research paper in high impact journal, and he has more than 1900 citations. And right now he is working as an assistant professor in Department of Emergency Medicine, University of Maryland, Baltimore, where his research focus on the understanding the molecular and cellular mechanism driving neuroinflammation, neuroimmune signaling, and cognitive dysfunction associated with toxic exposures and injury-related neurological disorder. So yeah, here we have Dr. Bhatt.
Thank you,
Yeah,
Sadiq, for
please go
this
ahead.
wonderful introduction, and thank you, Asset, for giving me this present to share the findings from our lab. So our lab basically works on a sickness, which I mean, a disease which most of you probably won't be knowing. So it's called decompression sickness. So this is a disease that people, usually the Navy divers as well as scuba divers, or in general, those people who take pressurized air. Uh, so what is DCS? It's also called the bends. DCS is also known as the bends or a barotrauma and refers to injuries caused by the rapid decrease in the pressure that surrounds you or either in water or air. So so there is DCS for those people who go underwater, and there's also aero decompression for people, especially for astronauts. It occurs mostly in scuba or deep sea divers, although it can occur during high altitude or unpressurized air travel. DCS has CNS manifestations with prevalence ranging from around 30% in compressed air divers to as high as 56 in breath hold divers. 30% in compressed air divers reported signs of DCS, and it's estimated that 3 cases per 1,000 dives in sports diving, especially in scuba divers. The risk of DCS is 2.5 times higher in males than in females. So what are the symptoms for DCS? As per the Divers Association Network, which is based in US, I mean, it's a global organization, but they have headquarters based in North Carolina. So around 68% of the patients, they report pain as all the symptoms. First symptom, and 40% of patients report first symptom as a pain. And there are other associated symptoms like numbness, dizziness, motor weakness, muscle discomfort, mental status, pulmonary. It affects almost all the systems in the body. So what usually happens is, when since you have the pressurized air, you're taking the air from the pressurized gas cylinder, but when you come, slightly as you come out of the water, there's a decrease in the gas pressure, which results, a gaseous nitrogen bubble is formed inside in most of the tissues. As such, we say that nitrogen is an inert gas, but it does trigger the reactive nitrogen species in oxidative stress, which results in the formation in the release of inflammatory mediators, and those mediators are responsible for are involved implicated in the DNS pathology. So it has two forms. One is type 1s that affect skin, musculoskeletal, and lymphatic system, whereas the second one, that's the type second, it affects the CNS, it affects cardiovascular and pulmonary involvement. So our lab focus on the delayed neurological sequelae. So these are among the most potentially and deadly outcomes of DCS. And the symptoms are chronic numbness or tingling, persistent weakness or partial paralysis, impaired coordination balance, chronic neuropathic pain. If they have attention deficits, they have memory disorders, they have motor dysfunction also, and bladder and bowel dysfunction. And also they are become prone to neurological subsequent exposures with this nitrogen or with the pressurized air. They make them prone to these neurological DCS. And there are studies where it has been reported that these scuba divers, they are highly susceptible to Parkinson's as well as Alzheimer's disease. So what we do is our lab, we focus because we look at the microparticles. These microparticles are the large extracellular vesicles released in the body. But why microparticles? Because the nitrogen bubble, which is formed because of the pressurized air, it dissipates quickly. But the DCS still persists. Even we have done studies where the nitrogen bubble will dissolve within a couple of hours, and you will still see the delayed neurological sequelae in these disease model, high pressure exposed mice till 3 weeks. So what is that drives this DCS DNS in decompression sickness? We found that these are the microparticles. So what are these microparticles? Microparticles are also known as microvesicles. These are the small vesicles that originate from the plasma membrane of almost all eukaryotic cells, and they are released during apoptosis or even during the normal activation. We call them FedEx. So they are the transporters inside the body, which their size range from zero point 0.1 to 1 micron, and they carry lipids, proteins, nucleic acids, small RNA, microRNA, and non-coding RNAs. Yeah, and they play an important role in the pathogenesis and subsequent progression of cardiovascular, metabolic, and neurodegenerative disease. So our lab has we have established that in DCS there is neuroinflammation with increased lymphatic flow, and we have studied both in mouse model as well as in the human blood samples which we receive from either from the Navy divers or from the scuba divers throughout the world. In clinical studies, in these blood samples, we check the neutrophil, what happens to neutrophil, or what are the changes that are taking in the blood. So we did see that high pressure exposure, it triggers the activation of neutrophils. And we also saw that there is an increased production of microparticles. There is higher levels of elevation of microparticles, which carry pro-inflammatory mediators as well as which carry CNS, which carry proteins that are specific to brain. Like we did find these microparticles expressing GFAP, IBA1, MBP, NPR. Most of they are all CNS specific, which indicates that these proteins are released in the brain and then they are liberated via lymphatics into deep cervical lymph nodes, and from there they come to the blood. So that's what we found out from our clinical studies, and then we try to recapitulate it in the mice model. We have a mice model where we try to expose the mice to high pressure at 100 psi for 2 hours so that we can mimic the conditions. Our lab, we have shown that there's a neuroinflammation with increased lymphatic flow in the murine model. It immediately happens after 2 hours, and it persists for, we have done the acute exposure and then we have done the study till 3 weeks. So we saw that there is a persistent neuroinflammation in the brain, there's a persistent neutrophil activation, and there is persistent increase in these microparticles in the blood as well as in the deep cervical lymph nodes. And it's worthwhile to mention that we also saw that the lymphatic system, that we did MRI as well as other fluorescent tracing images where we found that there is increase in the lymphatic flow. And then we also tried, we were looking, okay, what is a therapeutic alternative which we can use so that at least we can look for a therapeutic target. So we did a study on recombinant human plasma gel solvent. We injected these mice with the recombinant human plasma gel solvent, and they behave like normal. And luckily, this was, we got it funded, and this clinical trial is still going on where we are evaluating whether this recombinant plasma gel solvent protein, if given to the scuba divers, does it produce any anti-inflammatory response in these healthy divers or not? So what our hypothesis is, we say that high pressure exposure post decompression, because of the nitrogen bubbles, it triggers a nitrogen, it triggers reactive oxygen species, oxidative stress, lipid peroxidation, which results in the production of which results. So there's a first cycle of microparticles which are released from the brain. When there's a glial cell activation, there's an immune response in the brain. It triggers a release of inflammatory microparticles. And these microparticles, they go in the deep cervical lymph nodes, where they also trigger lymphocyte proliferation. This is liberated the lymphatics, and from deep cervical lymph nodes, they are released in the blood because they have these proteins, these microparticles. They carry TSP, they carry CD36, they carry other proteins which trigger the neutrophil activation. So in on neutrophils, they react with three receptor combinations called RAGE, CD36, and TLR4, which results in the additional production of microparticles. These microparticles are used by neutrophils. It contain effectin, Ly6G, and these additional production of microparticles, they go and interact. We hypothesize that they go and interact with the CD36 present. They go and bind with the CD36 present on the blood-brain barrier and thereby perpetuate the neuroinflammatory process. This is how this continuous vicious cycle of neuroinflammation goes on. Continuous neuro as well as systemic inflammatory response keeps on going even for 3 weeks. So as you can see here, there and plus they also we had done studies where we did see that post three weeks of the exposure, we saw that they have impaired neurogenesis also. But when we inject it immediately after the high pressure exposure, when we infuse LY recombinant human gel solvent, what it does is it lyses these microparticles, so which prevents neutrophil activation, which also impedes, which lyses the microparticles, prevents the neutrophil microparticles, and in turn prevents the BBB deterioration or impairment. And as a result, it blocks multiple cycles of neuroinflammation. That's how it should. And it also improved the. We saw that there is an improve. It didn't affect the neurogenesis, and there was no degeneration in the hippocampal region. And these mice also showed better attention performance as well as improved learning and memory. But we wanted to see, okay, so if these affected microparticles produced by high pressure exposure, do they really go on? Because we hypothesize that they go and bind with the CD36. To illustrate that, we use different strains of the mice. We use C57, we use total conditional knockout CD36, and we deleted CD36 on endothelial cells and then exposed them to high pressure, wait for 12 days, did our conventional MRI, and we also did cisterna magna imaging, euthanized, collected blood, lymph nodes for the flow cytometry for neutrophil activation as well as the microparticle production, and brain for histochemistry as well as Western blotting. So to confirm whether it's these microparticles which trigger DCS, we expose the mice to high pressure exposure, let it, sag the mice after 2 hours, collected the microparticles, purified them. And after purification, we injected these microparticles from the high pressure exposed mice into the naive mice. And then we put them back in the colony, waited for 12 days, then did the conventional MRI, whatever stuff we were doing. So after 2 weeks, we sack the mice, we collect the blood for the neutrophil activation for the microparticles. We did differential centrifugation and ultracentrifugation to purify the microparticles from the blood. And as you can see, we did the characterization via alloyed in MP to confirm whether it has effect in microparticles, whether it has effect in attached rate or not. And so for the gating purpose, we gated them based on the bead size. So we have a bead size from 0.3 micron to 1 micron. Yeah, that's how we did the gating, to differentiate whether they are small or they are large microparticles. So coming to the results, so activation, as you can see, that we did see that the neutrophils have higher expression of myeloperoxide, CD18, when you compare with the control. So these high pressure exposed NA mice had higher expression of MPO and CD18, which indicates that their neutrophils are activated. Similarly, as you can see here, the wild type NA mice, when injected with microparticles isolated from the high pressure exposed mice, they also had higher pressure and much higher neutrophil, much higher expression of MPO and CD18 when compared to the control mice, which indicates that their neutrophils are activated. But as you can see in the CD36, total knockout, they don't have any increase in the expression. It's not a significant difference. Even the total knockout with injected with microparticles, we didn't see any increase in the expression of myeloperoxide or CD18 or CD41. We did see an increase in we did see an increase in the neutrophil activation. We did see the increase in the markers for neutrophil like myeloperoxide, CD18, and CD41 in mice in endothelial deficient CD36 mice. This indicates that, okay, we did expect that they are going to have neutrophil activation because CD36 still does exist on the neutrophils. Similarly, we didn't see when we injected these mice with effectin, we didn't see any statistical difference in MPO and CD18. Then coming to the circulating microparticle count and subpopulation. So these are the lymph nodes where we pulverized them, isolated, then did the microparticle assessment. We saw that total microparticle count in deep cervical lymph nodes of high pressure exposed mice post 2 weeks is higher, significantly higher than compared to the control, both in wild type mice exposed high pressure as well as in those having injected with microparticles. But we didn't see in CD36 total knockout, we didn't see any difference in the endothelial deficient CD36 knockout. We also saw that subtypes, the CNS specific proteins expression on these microparticles, we saw that there's a higher expression of GFAP, there's higher expression of MBP, which might be responsible for the priming of lymphocytes in the deep cervical lymph nodes. There is higher expression of NPR, there's higher expression of TR, thrombospondin, there is higher expression of TSP, TMEM119, and a microglial marker. And we also saw that there's a dual expression of certain microparticles containing both TSP and GFAP. This all we saw an increase, and there's a statistical difference between the control as well as the disease and high pressure control mice. But we're surprised we didn't see any statistical difference increase in the expression of these microparticles from in total knockout, total CD36 knockout, as well as flocks conditional CD36 knockout, both high pressure exposed as well as those injected with the microparticles derived from the native mice, indicating that CD36 is necessary for the activity of microparticle or high pressure increased microparticle count. Then coming to the blood, we did the microparticle, we also checked that microparticle count in the blood where we saw that there's an increase as seen here. There is increase in the blood microparticle count, both in the blood as well as in lymph nodes, both in high pressure exposed as well as those injected with the microparticle in the native mice. And there is increase in LY60, CD36, and CD41. These are the blood-related microparticles, which we say that these microparticles, they originate from the blood, but some of them also expressed GFAP, MBA, NPR, TSM, and TMEM119, indicating that there is an insult going on the brain, which triggers the release of these microparticles from the brain, and then from brain they traveled all the way via lymph nodes and are released in the blood. And in the blood, they did trigger the production of the additional microparticles. But while as you can see here in the knockout mice, there is no significant difference. In fact, they had a lesser microparticle count when compared to the wild type control. Similarly, the flocks mice, total knockout mice, high pressure exposed flocks mice, we did see an increase in the blood microparticle subtypes in flocks. That's the conditional endothelial CD36 high pressure exposed as well as injected mice, but we didn't see any increase in the expression of CNS specific markers, which also indicates, which confirms that, okay, so they are triggering that neutrophils, no doubt they are activated. But because CD36 is deficient on the endothelial cell on the BBB, it doesn't elicit any further neuroinflammatory response. Then we did some imaging. We looked for the phosphodiesterone because we hypothesized that these microparticles they do have effect. They do have phosphodiesterone attached with them, which goes and binds with the CD36. Uh, so as you can see that there is an increase in the production of phosphodiesterone in the high pressure exposed mice, while as you see the high pressure flocks mice, the conditional there's yeah there's no increase. There's no increase. It also indicates that it doesn't go and bind with the vascular with the CD31 attached to the BBB or with other blood vessels. Then coming to the we also assess the lymphatic flow. We did hypothesize that maybe there, because of the high pressure, there's a vascular injury, and let's see what happens with the lymphatic flow. So we measured it via two ways. We injected the lymphatic tracer, as you can see here. We injected a CSF tracer. After exposure, we injected a CSF tracer. It's two dyes. One is cadaverine, and then there's an O-albumin, and then saw their distribution, diffusion inside the brain. You can see here, this is a wild type mice. You can see that there is increase in the cadaverine. Here is 100 psi. You can see that there is an increase in the cadaverine distribution inside the brain of the wild type mice, but this is the knockout and this is the conditional deficient mice. We didn't see any statistical increase in the tracer distribution in the brain, which indicates that there is an increase in the lymphatic flow. Similarly, we also, with the uh, O-albumin, we didn't see any statistical difference also, except we did see increase in the O-albumin, but it's not statistically different. Then we did a contrast-based imaging using the MRI. What we did is after exposing the mice to high pressure, put them back on the colony after 3 weeks, we we did the MRI. We injected this contrast gadolinium inside the tailwind and did a time. Uh, we did 5 minutes multi interval imaging, and we found that there is an increase in the there is an increase in the flow in the wild type mice, but in knockout you can see here there's no increase. It's like they behave normally. It's like conditional deficient. They also both as and we also saw the clearance, that the clearance is higher in um, in knockout while compared to control. So this also indicated to us that yeah, so it's a CD36 which is responsible endothelial CD36 which is responsible for maintaining the lymphatics in these DCS mice. Further, we also detected that whether there's any vascular injuries using a rhodamine labeled dextron. We infused the rhodamine inside in these mice after 2 weeks, and then we saw that there is a statistical increase in the fold in the rhodamine inside the brain as well as skeletal muscle, indicating that there is a vascular injury. That's why the rhodamine distribution is higher in the decompressed, as well as it mimics in wild type microparticle injected in mice. But you can see here in the control in CD36 knockout as well as in the injected mice and in the flocks mice, there is it's not higher, and in fact it's slightly lower, which indicates that it does prevent the vascular injury also in these DCS mice. Then we looked at the we did some Western blotting to look at the expression, evaluate the expression of certain inflammatory markers in the brain. So first we did the neutrophil-mediated immune response of the brain. We looked for the thrombospondin, myeloperoxide, LY60 actin, LY60, we saw that there is an increase in the expression. As you can see here, this is a wild type decompressed mice. There is higher expression of thrombospondin, myeloperoxide, as well as LY60, indicating that these neutrophils, they have started infiltrating in the brain and mediating the immune response in the brain. Then we also looked for the conventional astrocyte and glial cell markers. So we saw that there's an increase in the glials in GFAP. We also checked the expression of the IBA1 microglia and phosphorylserine and NFKB. So there is increase in all there's an increase in the inflammatory markers indicating that both astrocytes and microglia are reactive. Because they are reactive, they as a result they are producing additional microparticles and then released in the blood. So what we conclude from this study is that CD30, as you can see here, if there is CD36, the microparticles released during the DCS from the brain, they are come to the blood. From the blood, they react with the neutrophils, causing neutrophil activation, and these neutrophils neutrophil microparticles in turn carry effectin, TSP, CD36. They go and bind the CD36, this phosphorylserine on these microparticles, go and bind with the CD36 on the receptor on the BBB, which result in the lymphatic dysfunction, vascular injury, neuroinflammation, and in turn results in the cognitive dysfunction and attention deficits. While as in the flox mice, that's conditional deficit mice, there are CD36 knockout mice. So although high pressure exposure, it does trigger astrocyte and glial activation, which results in production of first cycle of microparticles from the brain. When they are liberated in the blood, they even the additional microparticles which are produced, they do have phosphorylserine, but because the receptor is not available on the BBB, they cannot go and bind with the CD36. As a result, it prevents the multiple neuroinflammatory cycles, and because of which via this we have indicated that there is a role of CD36 in the DCS model. And our lab is funded by NIH, and we get a consistent funding from Office of Naval Research. Our lab mostly works for the Navy, and we also get funding from the Emergency Medicine Association. Thank you. These are some references.
Yeah, nice presentation and wonderful work, Dr. Abid.
Thank you.
Yeah, does anyone have any questions? Okay.
Yeah.
Yeah.
So just I have a question that you are talking about the microvesicles, no? So and
Yes, sir.
at the same time, you're also talking about activation of neutrophils.
Yes.
So what does neutrophil play a role? Because macrophages and macrophages, they just phagocyte, what we say, any kind of debris, no?
Yeah.
So do the macrophages, they think that this is debris and they just engulf those particles?
So
and
what happens
at the same
is
time, neutrophils, because they, yes.
So what happens is because these microparticles, they carry the ligands, and these ligands, they will go and bind with the neutrophils, and they form there's a receptor combination called, it's CD36, RAGE, and TLR4. When these because they carry the ligands, they go and bind with these receptor complex, which further results in the additional production of these microparticles. That's what triggers the systemic inflammation.
Okay.
Yeah, and we did see
How about
there is an infiltration. So this we are still working on it, but it's almost done. So we have seen that because they also contain DAMP proteins, especially those coming out from the brain, they go and they do the lymphocyte proliferation, and there is increase in the T cell and B cell production in the lymph nodes. And these circulating T cell and B cell, they further go and they circulate from all the way. And because this BBB is already disrupted during this disease model, we are tracking them, and we see that the systemic ones, they go and directly go inside the brain and then perpetuate the neuroinflammation. So in short, it
Thank you.
forms a vicious cycle. So it starts from we're still trying to find out where does this originate? Does it originate in the brain, or it starts in the blood? But this cycle keeps on going. We are trying to stop this via using different methods to understand where does it take place. We try to lyse the microparticles to see what's happening, then try to use the inhibitors, which are only going to prevent the neutrophil activation, and then see what happens in the brain. And we also recently we did a surgery where we blocked the outflow from the brain, just block it via deep cervical ligations, lymph node ligations. And then we saw what are the changes taking place in the blood, what are the changes taking place in the brain. We are still studying this.
Thank you very much.